let's go over them. Regulation of gene expression. If you have a gene that constantly making protein molecules, what will happen? What do you think will happen? Constantly protein molecules are being made. You remember gene expression? We talked about gene expression. Now today we are going to talk about regulation of those expressions. If you have a gene that constantly makes protein molecules, what will happen? Cancer. The cells become full of protein molecules. Variety of things can go on. So a gene must be turned off and turned on. Off and on. A gene must be turned off and on. If it doesn't turn off and on, then one example you're familiar, I just give you a quick rundown, it's cancer. Okay, so bacteria, let's talk about gene regulations in bacteria, and then I will talk about gene regulations in eukaryotic cells. First bacteria, then eukaryotic cells. So let's go over bacteria gene regulations. Very simple, straightforward, 4 or 5 PowerPoints, and that's it. There is not much, there is more, but your textbook, and most textbooks do not spend too much time on prokaryotic cells gene regulation. Okay, so let's go over that. Bacteria often respond to environmental change by regulating transcriptions. And that is true for eukaryotic cell as well. Eukaryotic cells and prokaryotic cells respond to the environment. Okay, for example, when we all leave, what is the environment of the cell? What is the environment of the room? When we all leave the room, what is the environment of the room? Oh, empty, thank you. The environment of the room is empty, and then what will happen? The gene is turned off. And then when we come inside, from outside, we all accumulate outside, and then we come inside, what is the environment of the room? It's full, it's occupied. Then the gene must be turned on. So that is true both for eukaryotic cells and prokaryotic cells. The environment decides what genes must be turned on or off. Am I making some sense? So that's a very important concept. Environment. And of course, your textbook talks about natural selection, and that is something I have not talked about yet. I will toward the very last day of class. I will talk about natural selection and what Darwin proposed a little bit, so you can put all of that information together with what is have said so far. Natural selection has favored bacteria that produce only the products needed by the cell. A cell can regulate the production of enzymes by feedback inhibition, which I'll talk about that here in a minute, by gene regulations. One mechanism for control of gene expression in bacteria is the operon model. So operon model, it's something that we need to talk about, we'll hamper on, and we'll go over in a minute. Okay, so you will see this again. I'll show it to you, but please, for now, put it in your brain, put it somewhere, open up a little place in your brain right here, and put it there. And let's go over this. A precursor, some kind of molecule, with help of that enzyme, becomes another precursor, another molecule, eventually becomes a tryptophan. And these precursors, each one of them becoming another one, there is an enzyme involved. How many enzymes involved here? Huh? How many enzymes? 3, thank you. And then tryptophan, what is tryptophan? Anybody knows what is tryptophan? Semester is coming to the end. Huh? Well, it's an amino acid. It's an amino acid. At the beginning of semester, I said, get to know them. You need it for rest of semester. Here we go. You need them again. So tryptophan is an amino acid. The cells make them. We are talking about bacterial cells. Bacterial cells are making them, but they take other molecules and convert them eventually to tryptophan. Okay. This structure right here is a DNA or a gene. Gene, TRPE, they call it. TRPD, tryptophan. TRP is the abbreviation for tryptophan, amino acid, tryptophan. Okay, E, D, C, B, A. So A and B make up enzyme C. These are the genes. Am I making some sense? This is a gene. And a gene is DNA, right? I labeled them up there for you. If you want these final PowerPoints at the end, start studying these PowerPoints. But at the end, I'll give you the PowerPoints. The PowerPoints that I sent you last night, it did not have this label. I don't know. If you have the PowerPoint before you, you can label it right there. Okay. So, and then this gene makes up enzyme number 2. And 2 genes, 2 of these make up enzyme number 1. I hope I'm making some sense. So, 1 enzyme does not mean made by 1 gene. 1 enzyme can be made by 2 genes. 1 enzyme can be made by 1 gene. 1 enzyme is made up by 2 genes or more. Okay, I hope that's another thing. Now, what will happen? If there is ample tryptophan inside of the cell, remember the example, if we come inside of the cell, if we are leaving, we're all leaving this cell, this room, I turn off the lights. Is that right? That's the environment. But I'm saying, if there is ample amount of tryptophan inside of the cell, this tryptophan goes back and turn off that enzyme. When that enzyme is off, shut off, then that precursor does not become this one, that precursor does not become that one, and that one does not become tryptophan. But if there is not enough tryptophan inside of the cell, then this will take place. That's one way. They call it feedback inhibition. They're calling this feedback inhibition right here. The other way is by excess tryptophan. Excess tryptophan will go ahead and turn these genes off. Turn the genes off. This one we will not talk about. Negative feedback at the beginning of semester, I'll probably spend 1 minute or 2 minutes or 5 minutes on it. This one we are not going to talk about. But this one, the focus of this chapter is this. How to turn these genes off. And, of course, on. We'll talk about on and off, both. This chapter is just not about turning genes off. It's about turning genes on. If your genes are always off, there's no protein molecules being made. Nothing is being made. Okay, so let's move on. Operons, okay, that's a term you need to become familiar with. Operons, the basic concept, a cluster of functionally related genes, a cluster of functionally related genes can be coordinately controlled by a single on-off switch. I've been talking about that all morning so far. The switch is a segment of DNA called an operator, usually positioned within the promoter. The promoter we already talked about in the past. What is an example of a promoter? Can you give me Tata box? Very good. That was one example of a promoter. Multiple TA. Okay, so what am I saying? The operator is usually positioned within the promoter. And an operon, okay, that's something I'm hoping you would familiarize yourself with. An operon is the entire stretch of DNA that includes the operator, the promoter, and genes that they control. So operon is a term that is referred to the entire promoter, operator, and the gene that they are controlling with. Okay, let's go look at some pictures.
Can you go back one slide real quick? I'll ask you a question. What is the precursor?
A precursor, for example, a molecule inside of a cell, such as a another amino acid, such as lysine. Okay, so lysine, that would be a precursor, or
And then the
a
enzyme-
cholesterol,
And the
anything.
enzyme sits inside of them, or what?
The what?
The enzyme, what does it do?
Enzymes will turn that precursor, could be another molecule, any molecule, into another precursor, and eventually to tryptophan. So it takes a few stages
Oh, so the
of
whole,
chemical reaction for a precursor to become tryptophan.
so the whole thing is to get tryptophan?
Yes, the idea is to get the tryptophan.
Express enzymes.
Right.
Into
Turn, for example, cholesterol. If you are happy with cholesterol, it turns it into tryptophan.
the, okay. Alright.
Please ask. Okay. Okay, the operons can be switched off by protein repressors, and I will show you, we'll see some pictures of the repressors. The repressors prevent genes transcription. Gene transcription, you already know what that means. What is transcription? Refresh my memory. I forgot.
RNA synthesis.
RNA synthesis. Okay, so RNA synthesis by repressors, transcription by binding to the operator and blocking RNA polymerase. It blocks RNA polymerase is there inside of the cell, but it blocks. Okay, the repressor is the product of separate regulatory genes, which we'll see that. I don't think we have a PowerPoint on that one, but we'll see. The repressor can be in an active or inactive forms depending on the presence of the other molecules. A co-repressor, helps repressor, is a molecule, a co-pilot, you remember co-repressor is a molecule that cooperates with a repressor protein to switch an operon off. Okay, it's an off. For example, E. coli in synthesis of amino acid tryptophan, when it has insufficient tryptophan, that's a repressor. Oh, a lot of, in this chapter, it is a lot of writing. I didn't think that I should get rid of them for you guys, because in the past, students said this is the hardest chapter, but I don't think this is the hardest chapter. I think this is the easiest chapter. Just a few terminologies, new terminologies. By default, the tryptophan operon is an on, and the genes for tryptophan synthesis are transcribed. When tryptophan is present, it binds to the tryptophan repressor proteins, which turns the operons off. The repressor is active only in the presence of its co-repressor tryptophan, thus the tryptophan is turned off or repressed if tryptophan levels are high. Here we go. Great. So this is an example of, first of all, this is an example of a negative control. Okay, and then you will see an example of a positive control. It means the genes are being turned off. Okay, right here you have a promoter, DNA, you have a promoter, you have tryptophan, regulatory genes, and then downstreams right here you have the RNA polymerase, and right here is your promoter. This segment is promoter, and this is E, D, C, A, B, and A. And right here, you remember that? The last PowerPoints I showed you, A, B, C, D, E. That's what they are, right here. Okay, those are genes. Those are the genes that is responsible for one protein molecule, another protein molecule, another protein molecule, another protein molecule, another protein molecule. And between each one of these protein molecules, you remember the stop codon? UUA was stop codon. Start codon is AUG. Okay, so each segment is responsible for these protein molecules. But that's something you already know. What is it that you don't know is this part that we are talking about, the controlling part. This chapter is about this part. This part you already know. Okay, mRNA is being synthesized, 5' end to 3' end, RNA polymerase, this RNA polymerase is making up the RNA, all of that is known. But this segment right here, that you don't know, with the controlling gene, the turning off, I'm not going to turn off the switch of the electricity anymore. You already know that. Okay, so this is the switching off and on. Okay, let's talk about that. Well, since you already have that picture, I'm going to take this one, a picture, and enlarge it. All I wanted to show you on this one, that you know where these are coming from the last PowerPoint. The precursor, Brian asked, the precursor became another precursor, and eventually became tryptophan, right? So I put it in here that you guys know what we are referring to, right here. Okay, so let's look at the first one. Right here, I'm not too worried about it. This is the part that you should worry about, right here. So you have a protein molecule, it's called inactive repressor. This is inactive, this protein molecule is called inactive repressor. And tryptophan R makes the RNA, and that RNA is making that protein molecule. So this is a repressor that is inactive. It means when this is inactive, what will happen? It means these protein molecules are being synthesized. Do I make sense? RNA polymerase will do its job when this is inactive. Okay? And that's why I say operon on. The operon, the switch is on. The power switch is on. The operon is on. This is inactive. The repressor, repress, is inactive. So operon is on. And when operon is on, when this is on, when the whole entire thing is called operon, when this entire thing is on, then of course, right here, protein molecules are being synthesized. Now, what will happen if there is excess tryptophan? That tryptophan is called co-repressor. That tryptophan will sit right here into the protein molecules, the repressor. This is the repressor protein molecules. And tryptophan sits right here, and then this repressor becomes active. That repressor will attach to that operator right here. This is the operator. When it attaches to the operator, then the RNA polymerase cannot proceed. It's blocked. Do I make sense? This is how controlling of a gene, of course, very simplified version of it, happens. This is how genes are being controlled. Okay, so this is operon off, operon on. Okay, I hope excess, let's go back, excess tryptophan, turn off the gene. Excess tryptophan, turn off the gene. I'm not talking about the enzyme. That's a different story. We learned that at the beginning of semester. Okay, oh, sorry. Okay, let's start doing the lab because I want you to go over the quiz. So I give you a prelude to the chapter.